Parietal cells, also known as oxyntic or delomorphous cells, are the epithelial cells that secrete hydrochloric acid, HCl, and intrinsic factor. These cells are located in the gastric glands found in the lining of the fundus and in the body of the stomach. They contain an extensive secretory network, called canaliculi, from which the HCl is secreted by active transport into the stomach. The enzyme hydrogen potassium ATPase, H+/K and ATPase, is unique to the parietal cells and transports the H and against a concentration gradient of about 3 million to 1, which is the steepest ion gradient formed in the human body. Parietal cells are primarily regulated via histamine, acetylcholine, and gastrin signaling from both central and local modulators. See regulation. Secretion of hydrochloric acid. Synthesis. Hydrochloric acid is formed in the following manner. Hydrogen ions are formed from the dissociation of carbonic acid. Water is a very minor source of hydrogen ions in comparison to carbonic acid. Carbonic acid is formed from carbon dioxide and water by carbonic anhydrase. The bicarbonate ion, HCO3, is exchanged for a chloride ion, Cl, on the basal side of the cell, and the bicarbonate diffuses into the venous blood, leading to an alkaline tide phenomenon. Potassium, K+, and chloride, Cl, ions diffuse into the canaliculi. Hydrogen ions are pumped out of the cell into the canaliculi in exchange for potassium ions, via the H+/K and ATPase. These receptors are increased in number on luminal side by fusion of tubulovesicles during activation of parietal cells and removed during deactivation. This receptor maintains a million-fold difference in proton concentration. ATP is provided by the numerous mitochondria. As a result of the cellular export of hydrogen ions, the gastric lumen is maintained as a highly acidic environment. The acidity aids in digestion of food by promoting the unfolding, or denaturing, of ingested proteins. As proteins unfold, the peptide bonds linking component amino acids are exposed. Gastric HCl simultaneously activates pepsinogen, an endopeptidase that advances the digestive process by breaking the now exposed peptide bonds, a process known as proteolysis. Regulation. Parietal cells secrete acid in response to 3 types of stimuli. Histamine stimulates H2 histamine receptors, most significant contribution. Acetylcholine, from parasympathetic activity via the vagus nerve and enteric nervous system, stimulating M3 receptors. Gastrin, stimulating CCK2 receptors, least significant contribution, but also causes histamine secretion by local ECL cells. Activation of histamine through H2 receptor causes increases intracellular CAMP level, while ACH through M3 receptor and gastrin through CCK2 receptor increases intracellular calcium level. These receptors are present on basolateral side of membrane. Increased CAMP level results in increased protein kinase A. Protein kinase A phosphorylates proteins involved in the transport of H+/K and ATPase from the cytoplasm to the cell membrane. This causes resorption of K and ions and secretion of H and ions. The pH of the secreted fluid can fall by 0.8. Gastrin primarily induces acid secretion indirectly, increasing histamine synthesis in ECL cells, which in turn signal parietal cells via histamine release/H2 stimulation. Gastrin itself has no effect on the maximum histamine-stimulated gastric acid secretion. The effect of histamine, acetylcholine, and gastrin is synergistic, that is, effect of 2 simultaneously is more than additive of effect of the 2 individually. It helps in nonlinear increase of secretion with stimuli physiologically. Intrinsic factor. Parietal cells also produce intrinsic factor. Intrinsic factor is required for the absorption of vitamin B12 in the diet. A long-term deficiency in vitamin B12 can lead to megaloblastic anemia, characterized by large fragile erythrocytes. Pernicious anemia results from autoimmune destruction of gastric parietal cells, precluding the synthesis of intrinsic factor and, by extension, absorption of vitamin B12. Pernicious anemia also leads to megaloblastic anemia. Atrophic gastritis, particularly in the elderly, will cause an inability to absorb B12 and can lead to deficiencies such as decreased DNA synthesis and nucleotide metabolism in the bone marrow. Canaliculus. A canaliculus is an adaptation found on gastric parietal cells. It is a deep infolding, or little channel, which serves to increase the surface area, e.g. for secretion. The parietal cell membrane is dynamic. The numbers of canaliculi rise and fall according to secretory need. This is accomplished by the fusion of canalicular precursors, or tubulovesicles, with the membrane to increase surface area, and the reciprocal endocytosis of the canaliculi, reforming the tubulovesicles, to decrease it. Diseases of parietal cells. Peptic ulcers can result from overacidity in the stomach. Antacids can be used to enhance the natural tolerance of the gastric lining. Antimuscarinic drugs such as pyrazepam or H2 antihistamines can reduce acid secretion. Proton pump inhibitors are more potent at reducing gastric acid production since that is the final common pathway of all stimulation of acid production. In pernicious anemia, autoantibodies directed against parietal cells or intrinsic factor cause a reduction in vitamin B12 absorption. It can be treated with injections of replacement vitamin B12, methylcobalamin, hydroxycobalamin or cyanocobalamin. Achlorhydria is another autoimmune disease of the parietal cells. The damaged parietal cells are unable to produce the required amount of gastric acid. This leads to an increase in gastric pH, impaired digestion of food and increased risk of gastroenteritis.
Please subscribe and thanks for watching.